I come from SATEC BUT, that is Brno University of Technology. It is institute scientifically oriented and is a consortial institute with other universities in Brno. In my group, I lead the Experimental Biophotonics Group at this institute, and one of our most important and long-term project is just the optical setup, which is the basis of a Q-phase microscope.
How did you come up with the idea for the Q-phase microscope?
In the 90s, I thought about the possibility to build a confocal microscope without scanning. And I was very intrigued by the idea of Emmett Lathe, which was one of the founders of Axis holography, on equivalence of the holographic system with incoherent light source and the confocal imaging process. So we developed this idea and built an optical system of holographic microscope which could work completely with completely incoherent source, and this was the basis of Q-phase.
What is the difference between coherent and incoherent light?
Coherent light is the light from laser. It's not very natural light, and this light has many interesting properties like diffraction or interference, which is not natural for human being. The normal light from the sun or from the light bulb is incoherent, and this is normal light for us. So for wide field microscopy, it's much more better to use incoherent light because the image is very clear and naturally looking.
Can you give us a short description of the Q-phase light path that we see here?
Microscopes we design actually consist of two microscopes with condensers, objective lenses, detector in the output, and the table of the specimen. Here in the Q-phase and our other designs, these two microscopes serve to compare the signal which is influenced by the specimen with the signal which is untouched by the specimen on the holographic plate. Properties of light carry the important information about the specimen, about the mass density of the living cell we would like to discover and to measure. For the observation of the specimen of biological nature, it is quite fine that we can choose one of several objectives in the turret. For example, we can use low magnification objective to have some context of the field of view, and then use high magnification, high NA objective to observe some details, for example, behavior of individual cells or of some subfield of the field of view.
What do you think Q-phase can contribute to the various tools that we have in terms of microscopy?
I see it. When observing living cells, by ordinary techniques, you usually label these cells with thumb dial, and you need to use very intensive laser radiation to illuminate and to image this, for example, living cells. But the intensive laser radiation induces phototoxicity that damage the cells, and also that shifts the experimental results.
Can you describe this image that we see here that came from the Q-phase system?
In this picture, you can see the Q-phase observation of the array of plasmonic nano-antennas, which can serve as a building blocks of metasurface optical elements. And Q-phase gives us the possibility to observe individual plasmonic nano-antennas and to measure the phase shift. On the right, this is the reconstruction of the red blood cell refractive index distribution in 3D, which was reconstructed for the optical sections recorded by Q-phase. So it's the experimental data which was processed, and we get the usual distribution of the refractive index of red blood cell.
What's fascinating about the QPI technology? Are there some unique features?
The unique feature of Q-phase follows the use of incoherent light. That means the image is artifact-free. There is no coherence noise in the image, so you also can get the measurement with unprecedented accuracy. We reach the accuracy of 7 femtogram per square micrometer of dry mass density measurement, and you also get better resolution, which is normal for incoherent light wide field systems. And in addition, when observe 3D specimen, you get optical sectioning capability, which gives you the possibility to observe, for example, cell inside a collagen gel block, and this is simulation of natural environment of organism.